Okay, hello everyone and welcome to BrainMap. The seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping housed in the Martinos Center. It's my great pleasure to introduce our speaker today, Dr. Catherine Lebel. Dr. Lebel is an associate professor of radiology at the University of Calgary and the Canada Research Chair in Pediatric Imaging. She leads the Child Brain and Mental Health Program at the Alberta Children's Hospital Research Institute and the Hodgkin's Brain Institute. Dr. Lebel received her PhD in biomedical engineering from the University of Alberta and completed her postdoctoral training in neurology and pediatrics at UCLA. Her research uses MRI to study how brain structure and function change with age in typical children and those with neurodevelopmental disorders, including fetal alcohol spectrum disorder and learning disabilities. She also examines how brain structure and function are related to cognitive, behavioral, and environmental factors, including the prenatal environment. I would just like to remind the audience to please address any question you have using the Q&A box or raise your virtual hand. Dr. Lebel, thank you so much for coming here today. The virtual stage is all yours.
Thanks so much for that introduction, and thanks for the invitation to speak today. So I'm going to talk to you today about brain injury in kids. And what I'm going to do is first give just a brief intro to that. Then I'm going to talk about some of the data considerations that we need to keep in mind when we do this type of research, but honestly, any type of imaging research. Then I'm going to talk about outcomes at the post-acute stage. So this is about 10 days following the brain injury. Then we'll get into some chronic and longitudinal outcomes, and I'm going to finish up by looking at how we can consider symptom status of the kids when we look at those outcomes. Excuse me. So traumatic brain injury is a substantial public health problem, and injuries affect millions of children each year. Fortunately, most of the cases of brain injury are mild. About 3/4 to 90% of them are mild. And they can lead to a variety of symptoms, including things like headaches, irritability, forgetfulness. Most of those symptoms resolve relatively quickly, again, fortunately for those kids, but about 1/3 of kids can have symptoms that persist for a month or even longer. So obviously those are concerning, and especially so because all of this happens in the context of a developing brain. So the brain is changing, of course, throughout childhood, and this injury can impact it differently at different times, but also the longer these symptoms persist, potentially the more they can affect that brain development as well. There we go. So when an injury happens, it induces these rotational and shearing forces in the brain, which can lead to axon injury. And this probably underlies a lot of the negative outcomes that we observe following brain injury. So, you can look at this with conventional imaging, but conventional images don't tend to show a lot of changes that are apparent. So we need to use more advanced and more quantitative methods to look at the time course of injury here. So one of the nice ways to do that is with diffusion imaging, especially when we're interested in axon injury. And this has been relatively widely used to look at brain injury already, but the findings are pretty mixed, probably because a lot of studies have had small samples. They have different timing following the injury, and we know that brain injury will change the further you get away from the time of injury. And so these studies are really heterogeneous with respect to what they looked at and what they found. So what's really needed in this brain injury research space are large longitudinal studies that are able to use advanced quantitative metrics and that have appropriate comparison groups so we can really understand what we're looking at and interpret that in the appropriate context. So this is really the motivation behind this large study called Advancing Concussion Assessment in Pediatrics, or the ACAP study. And this is a study led out of Calgary by Dr. Keith Yates. It's a cross-Canada study that recruited kids at 5 different sites: Calgary, Ottawa, Montreal, Edmonton, and Vancouver. And kids there were recruited in the emergency department either following a mild traumatic brain injury or following an orthopedic injury. Okay, an MRI was conducted as part of this study at the post-acute visit, so that's about 10 days following the injury, and then kids were randomized to receive a second MRI scan at either 3 months or 6 months. So this is a longitudinal study, but they didn't receive 3 time points of imaging. We have 3 time points of imaging data, but only half the sample at the 3-month and the 6-month time point. This is just a diagram of the study recruitment. So again, they were recruited in the emergency department, came back for a post-acute visit, as well as a 3-month and a 6-month visit, but only received imaging at one of those time points. And we have a total of almost 1,000 kids, with about 2/3 of those being mild traumatic brain injury and 1/3 being the orthopedic injury controls. We're also fortunate that the protocol is the same one that I've been using in an ongoing study we have of typical brain development. And so from Calgary, we have a third group of typically developing controls that can form sort of a second control in addition to those orthopedic injury children. So here we have 39 uninjured, typically developing kids from 8 to 15 years. So same age range, same protocol in Calgary. And you'll see some of the data I present today uses this as a third or as a second control group, but as a third group. Some of it doesn't and uses the whole ACAP sample. So when we're talking about any pediatric research, I think there's a lot of other factors we need to consider when we sort of acquire the data and when we interpret the data. And one of those is participant motion. So this is common in pediatric studies especially. And of course, it can affect segmentation, it can affect our quantitative metrics. And importantly, it can obscure the relationships that we're interested in looking at. I'm sure you've all seen lots of different types of motion, but I'm just showing a couple of examples here on the top from a T1 weighted image in this data set where we essentially see this ringing, particularly near the top of the brain, and that prevents us from doing automated segmentation of structures. And then in a diffusion weighted image below here, where we get this sort of Venetian blind artifact with a lot of signal dropout, that's obviously going to prevent us from getting accurate measures of FA and MD and accurate reconstructed tracts. So we work really hard in Calgary, and I know most studies do, to reduce motion at the outset from the beginning to prevent this. So in Calgary, one of the things we have is a scanner that looks like a rocket ship, and we have a mock scanner as well that looks like a rocket ship where we can train the kids and get them to practice. So this helps us reduce motion in the first place, but of course, motion's not going to be eliminated, right? Motion is still there, and even small amounts of motion can affect our segmentation and quantitative metrics. So in general, previous studies have shown that motion tends to decrease with age, so older kids are better able to stay still, and that motion is higher in clinical populations like children with autism spectrum disorder or attention deficit hyperactivity disorder. So we can, hypothesize that it might be different in kids with mild TBI, but there hasn't really been research done in this area. And in particular, it's really important for us to understand whether motion differs in our MTBI group and in the orthopedic injury group, which we're using as controls, so that we can appropriately interpret our findings and our data. So we looked at this ACAP data and we looked at motion in it. So we quantified motion in both the T1 weighted images and the diffusion data, as well as in our typically developing controls. For the T1 weighted data, we manually checked all of the images and they were rated on a 3-point scale. But we also used an automated method to quantify motion, which is extracting the Euler number from the free surfer pipeline. So this provides a quantitative estimate of motion. The values are negative, but higher values are closer to 0 indicate less motion. And this was correlated really well with our manual ratings. So we did this manual and this automated method, but I'm going to show you the automated results today. In the DTI, We also looked manually at all of the volumes, and we evaluated which volumes were motion corrected. And so this was done by a number of different raters, with a third rater in the case of disagreement. And when we did analysis, we removed the motion corrected volumes. But for the purposes here, I'm going to report the number of motion corrupted volumes, excuse me, and contrast that between the groups. We also had a automated method for the DTI, which is the root mean square displacement from the FSL diffusion toolbox. I'm not going to show those results here, but they agreed very nicely with our manual ratings of the DTI. So here we have the average Euler number in the T1 weighted data versus age. And these are the three different groups: the typically developing, the orthopedic injury, and the mild TBI. So right away, we can see that this is positively correlated with age, meaning that there's less motion in the older participants. And also we found less motion, so higher numbers or less motion in the females. We didn't find a difference between the three groups. Okay, the three groups all showed similar profiles of motion for this T1 weighted data, but we have more motion in the younger kids and more motion in the boys. Looking at the diffusion data, here is the number of bad volumes within the DTI data set. So lower numbers are better, that means less motion. And here we again saw this age-related relationship where older participants had less motion. We didn't actually see a difference between males and females for these number of bad volumes, although there were more males with really high motion. But here we did see a difference between the typically developing controls and the injury groups together. So the typically developing controls had less motion than both of our injury groups. So we find overall motion is worse in boys, it's worse in the younger kids, and it's worse in the two injury groups compared to our typically developing controls. But good news for our interpretation of the brain injury versus another type of injury, motion seemed to be similar in both of the injury groups, the brain injury and the orthopedic injury. This is consistent with findings from my own data in younger kids, for example, here. Now note, this is the number of volumes remaining, so higher is better. But we also see an age-related relationship with motion in younger kids, and prior studies have shown similar age-related relationships and similar higher motion in boys, as well as those other clinical groups. So overall, our results are really consistent with other research into scan motion and kids, and suggest that the orthopedic injury group that we have is a better comparator for mild traumatic brain injury kids. Certainly a more conservative comparison than a typically developing control group. The other thing that is really important to consider in studies like this and many other studies is that we often collect data from multiple different sites. And this is, of course, because it enables us to get much larger samples when we're collecting data simultaneously in other places. But this introduces variability into our data. So much like motion, we try to mitigate this on the front end by matching protocols as well as we can, but these types of differences still persist. So these are just a couple of examples of site variations in the ACAP data, and then in a different data set here, we've got modularity in the structural connectome, and you can see quite a bit of systematic variation by site here. Interestingly, the 3 GE sites and the 3 Siemens sites look quite a bit more similar, but we still have variation. This is a different data set looking at actually the same participants scanned at 3 different sites, and at each site they were scanned twice. So we see, again, some systematic variation here with one of the sites having lower values of white matter volume. So we need to deal with this appropriately so we can look at our measures of interest in our analysis. We've been using COMBAT, which many of you are probably familiar with, but this is a harmonization technique that has been shown to work very well for multiple different kinds of imaging data. And in particular, it's good because it eliminates the site variation that you see, but it preserves the desired variation, for example, by group or by sex or by age. And so we have a lot of different analyses we want to do in the ACAP cohort. One of them is on the structural connectome measures. But combat hadn't been evaluated for this, so we started by testing that in our ACAP sample and looking at how well it worked. And we looked at how well combat worked to harmonize our data both at the matrix level, so connectivity weights in the structural connectome, and at the parameter level, so on those final parameters that we extracted from that matrix. And so here on the left is our data before harmonization. And I've just shown efficiency here, but the metrics were all showed very similar behavior during the harmonization process. So we can again see systematic site variation here, and almost every pair of sites was significantly different from each other. When we did harmonization at the matrix level, it really attenuated those site differences, but some of them still remained. About a quarter of the site differences that we had without harmonization remained when we did harmonization at the matrix level. When we did harmonization at the parameter level, all of the site variation was eliminated. So this is really encouraging from an elimination of the site variation viewpoint. But of course, we also wanted to make sure that this wasn't eliminating the variation that we were interested in and that we wanted to preserve. So we looked also at group differences and age-related changes. When we looked at group differences, there weren't any group differences at any of the stages. So that wasn't, I mean, that was nice that group differences were similar across harmonization stages, but it's hard to convince yourself that we're preserving variation of interest when we don't see differences. So we looked at age, which we know many of these parameters change with age. And before harmonization, we do see a bit of a correlation with age, although it gets strengthened quite a bit when we do harmonization at either the matrix level or the parameter level. And of course, it's stronger at the parameter harmonization level. So really encouraging for the use of combat to harmonize the structural connectome data, and encouraging for our analysis of the ACAP data in this way. So I've highlighted for you a couple of data considerations, and now I'm going to show you some results from the ACAP data. So as I mentioned before, we get these rotational and shearing forces during a brain injury, which can lead to axon injury. But previous findings in pediatric brain injury are mixed. Overall, in the diffusion space, we tend to see more studies reporting higher FA and lower mean diffusivity following the injury, but that's not universal. There are other studies that find no differences, and some studies find the opposite. There haven't been any studies in the pediatric space looking at naughty metrics, but adult studies tend to show lower neurite density index following injury. And then finally, coming to sort of cortical changes, these findings have been really mixed in the literature to date. And as I said before, this is probably due to the variable timing post-injury. It's possibly due to the use of different comparison groups. So I talked a bit about how motion is different in the injury groups compared to the typically developing group. And of course, there's other methodological variations. So we have this need for large longitudinal studies like the ACAP study. As I said, this all happens in the context of the developing brain. And this is really important to understand as we sort of move into the results and as we try to interpret them when we have this known age-related variation layered on top of the heterogeneity of the brain injury itself. So we know from a growing and large body of literature that there are widespread diffusion changes throughout childhood and adolescence. And specifically, we see increasing FA. This is a younger age group and this is an older age group, but we see very robust increases of FA across the brain in childhood, and these tend to slow down during adolescence, but do continue, particularly for frontal and temporal regions, even into young adulthood. At the same time, we see decreasing mean diffusivity, again, very robustly across childhood, slowing down a bit during adolescence and early young adulthood, and eventually plateauing, but often not until young adulthood in many of the areas. We see some sex differences in diffusion parameter changes, but they're relatively limited, actually. Now, when we do see them, we tend to see faster or sort of more mature patterns of development in girls compared to boys. We have also looked at changes in naughty measures to characterize white matter development. And here what we see are, again, very robust correlations between neurite density index and age for most of the tracts across the brain. And in fact, the relationship between NDI and age is stronger than the relationship between FA and age. We don't see any changes in orientation dispersion index, so that remains pretty flat across childhood and adolescence. And then I want to touch on cortical changes as well, because I'm going to talk about some of the cortical changes following brain injury. And here I've shown a bunch of different examples. This is an example of subcortical volume in the hippocampus and cortical volume in the cingulate gyrus. We see increases across early childhood, but then we do see sort of a peak plateau and then some decreases later on. And this is borne out when you look at older participants here. So for example, this is thickness in the parietal lobe, and this is surface area in the parietal lobe. So quite robust decreases of cortical thickness with age across childhood and adolescence. And these are subcortical volume changes a little bit later in childhood and adolescence, where some areas are not developing so much and some are increasing a little bit more. So overall, we see increasing volume in early childhood and then decreasing a bit later, decreasing cortical thickness over childhood and adolescence, and larger brain volumes in males. So we do see much more robust sex differences when we look at gray matter, but again, a little bit of a hint that there's earlier development in females. So this is all kind of to keep in mind as we go through the brain injury results. Now, the ACAP study acquired 30 directions at a b-value of 900 and 30 directions at a b-value of 2000, 2.2 millimeter isotropic resolution. The other parameters varied a little bit by site and scanner, but these parameters were consistent. And then we acquired T1 weighted imaging with 0.8 millimeter isotropic resolution. We have this typically developing control group in Calgary with the same data. And then we used combat harmonization to harmonize data across sites before analysis. These are the numbers of kids that came for that baseline MRI at 10 days post-injury. So we ended up with about 500, just over 500 that passed our quality assessments. That was looking at motion, for example, with again, 1/3 of those being orthopedic injury and about 2/3 of those being mild traumatic brain injury. So I'm going to start with the diffusion findings here. And I think first, the most important thing to say is that we didn't find any global changes. And in fact, most of the areas we looked at with tractography did not show changes. We only found 2 out of the 10 tracts we looked at that had differences between the mild traumatic brain injury group and the orthopedic injury group. And here I'm showing those 2 that had differences. So in the superior longitudinal fasciculus, shown in red, we found higher FA in the brain injury group compared to the orthopedic injury group. And this is consistent with the majority of previous findings, though as I said, they've been mixed before. And then we found higher MD as well in the anterior thalamic radiation here in the brain injury compared to the orthopedic injury group. These tracts are important for a lot of different functions, but among those are things like executive function and attention, which are related to some of the symptoms that kids might experience post-concussion. So, these changes may underlie some of the symptomatology we see. But importantly, these are relatively localized changes, right? We're not seeing major changes throughout the brain following this type of mild brain injury. We also looked at naughty metrics, and here we didn't see any significant differences between the groups. We did, though, just for a sanity check, look at relationships with age, and we see very much what we would expect in typically developing kids here. FA increasing with age, mean diffusivity decreasing with age, and neurite density index increasing with age. No differences in these age-related changes between the two injury groups. So they appear to be having the same type of development here. We looked at the structural connectome as well, and I had talked about this a little bit in the data harmonization. And here we see, again, no differences between the injury groups. So I've just shown clustering coefficient and local efficiency here, but the OI and the MTBI groups looked identical. But interestingly, we did see differences between the typically developing controls and both of the injury groups here. So the typically developing controls have a higher clustering coefficient and they have higher local efficiency than both of those injury groups. Similarly, we see a number of brain regions with nodal differences in connectome measures. So for example, degree centrality and efficiency were lower in both injury groups in the fusiform gyrus, and the clustering coefficient was higher in both injury groups in a number of other regions shown in red. And I think this helps highlight, again, the importance of the control group. So if you're looking at brain injury compared to typical development, you're going to find a lot of differences here, but they're probably not due to the brain injury itself. They seem to be either related to a more systemic response to an injury of any kind, or perhaps they're related to some premorbid factors that are similar in both of the injury groups and maybe predispose those kids to some kind of an injury. But of course, even though these results aren't specific to the head injury, we do see a lot of changes in frontal and temporal cortex, which we know from other studies is the most affected here. And these regions underlie a lot of those symptoms that we do see following injury. So we also looked at cortical morphology using that T1 weighted data, and we looked at subcortical volumes. We saw almost no changes in volume, but we did see changes in the pallidum. And here the brain injury group had lower volumes than the orthopedic injury controls, but there were moderations by sex. So in the left hemisphere, only the males showed differences. In the right hemisphere, both of the groups showed reductions, but we're seeing probably a slightly stronger effect in the males overall than in the females. Cortical thickness showed more widespread alterations, but these were also moderated by sex and actually also by age, just to make it very confusing and complex in terms of the relationships we're seeing. So here we see differences in the same area, but the effects were again stronger in the males with more, more thinning. But then here in a couple of other regions, we see effects in the females that we don't see in the males. In terms of age, again, we saw different results. Here in this occipital region, we were seeing changes in older kids where the mild TBI group had a thicker cortex than the orthopedic injury group, which we did not see in the younger kids. So we're seeing clearly some moderations by sex and age, which is not surprising given differences in brain development here, but we're definitely seeing that these are important factors to consider when looking at brain injury. The other thing to consider is the time post-injury, of course. And so this was all of the data I've shown so far has been at 10 days post-injury, which we would call the post-acute phase. We also wanted to look longitudinally and see how these brain differences potentially changed or evolved over time. So these are longitudinal results from the same data that I've shown you previously. So I showed you this anterior thalamic radiation, this increase in MD that happens early post-injury. We see also higher MD at about 3 months post-injury, but then interestingly, by 6 months, this has reversed. So we can see a time course here that really shows differences at different time points post-injury, and probably reflects really how the brain is healing or trying to heal from the injury. We also saw some areas that didn't show differences initially have differences that popped up later. So for example, in the uncinate fasciculus, we did not see any changes at our baseline scan 10 days post-injury, but now we see higher FA in the TBI group at 6 months, and we see higher NDI in the TBI group at 3 months. So differences really appear to be kind of emerging over time in some of these areas. These are just a couple of other examples. The arcuate, we're seeing these emerging differences in FISO, which is the isotropic fraction of water from noddy. And we're seeing also differences emerging around 6 months in the cingulate. Of course, these also interact with age and sex. And so for some areas, we saw very complicated interactions. This is the cortical spinal tract. Here we see differences in older females at 3 months, and then older males at 6 months in FA. With NDI, we see some differences in younger females and then in older males, again, emerging as we go on. But I think the sort of consistent theme here is that some of these differences more of these differences are apparent at later time points than are apparent 10 days following injury. We see really complicated interactions with cortical thickness primarily, but with age and sex and time post-injury. So you can see again, some differences really emerging at 3 and 6 months that were not apparent at baseline. Okay, so we're seeing things pop up here. We see differences between the females and the males, sometimes actually findings in the opposite direction. And then we see differences between younger and older kids, again, sometimes with differences actually in the opposite direction based on age or sex. So, I mean, it's complicated to interpret this, especially with the really complex interactions. But I think we can pull out a couple of things. One is that we're seeing differences emerge over time. And two is if we consider brain development here, we know that cortical thickness decreases over time, and we know that that happens a little bit earlier in girls. So we have a little bit more mature brain structure in the females, and we have more mature brain structure in the older individuals. And if we look at the females and we look at the older individuals, we're seeing more thinning in that TBI group compared to the orthopedic injury group. So perhaps suggests that these folks with a more mature brain pattern are having almost accelerated thinning effects following the brain injury. Whereas the males and the younger participants are having perhaps a delayed thinning where the cortex looks thicker. Now, the other piece that is really important to consider here is symptom status. So mild TBI is associated with a lot of different types of symptoms, and most of these resolve by about a month. But in 30% of kids, these will persist. And it's really logical to think that the kids with symptoms may have different patterns of brain recovery than the kids without symptoms. So we measured symptoms in these kids following their injuries, and we were able to subdivide the mild TBI group into those with persistent symptoms at 1 month post-injury compared to those without persistent symptoms. And we found some differences based on symptom status. And of course, like the other results I've shown you, these were also moderated by age. So these are the tracts that initially we found differences in, and what we saw when we divided by symptom group is that the TBI group with persistent symptoms had lower FA than the non-persistent symptom group. Interestingly, the non-persistent group was also different from the orthopedic injury controls. So the persistent and the orthopedic injury groups actually looked more similar. So it's a little bit surprising to us, but it may suggest that the higher FA in this non-persistent symptom group reflects sort of a brain reserve or a higher brain resilience, perhaps, that helps some of these kids recover a bit more quickly following injury. In the thalamic radiations, we saw higher MD in the persistent symptom group in the younger kids, and lower MD in the persistent symptom group in the older kids. So again, important to interpret this in the context of age here, but the symptoms definitely seem to be associated with a very different brain profile, particularly in these younger children. Just if we're thinking about sort of brain development here, a reminder that we see FA go up with age. So the higher FA in the non-persistent group here, we generally associate with a more mature pattern of brain structure, whereas the higher MD we see in the persistent symptom group, we would generally associate with a lower or a pattern of a less developed pattern of brain structure. We also saw differences based on symptom status when we looked at pallidum volume. And here we found higher volume in the pallidum in males with persistent symptoms in both the right and the left, and in the females, slightly lower volumes in the right, but what seems to be a larger effect in males compared to in females. Okay, so to summarize what I've shown you, we see overall a lack of global alterations in connectivity and in volume and in thickness. So really, we didn't see any changes on a global level, but the changes we did see were more localized. And particularly, they were occurring primarily in frontal and temporal regions, although we did see some areas of change elsewhere. The changes we see are less at baseline and are more present at 3 and 6 months. So they appear to kind of emerge over time. And they do seem to be different in more mature versus less mature brains. And then finally, we've shown patterns of brain differences that vary by symptom status, with generally the kids with persistent symptoms appearing to be more affected in terms of their brain changes. So to kind of wrap everything up, we know that mild TBI is common in children, and we've shown here that it can lead to subtle brain changes up to 6 months post-injury. We've also shown very complex interactions between injury, time post-injury, age, and sex. So really important to consider all of these when interpreting the data. And it's important to consider heterogeneity following the injury, such as symptom status, when looking at the brain differences. But some of this heterogeneity that we're showing and seeing might help explain the discrepancies seen in prior studies, especially when we're talking about relatively small studies with only a single time point post-injury. So that's all I have for you today, but I want to say a big thank you to Keith Yates, who leads the ACAP study. So I'm involved in the imaging portion, but there's a lot more data there that they're digging into in other ways. I want to say a big thank you to Ayushi Shukla here and Ashley Ware here, who did most of the analyses I showed you today. Lisa Marie Langevin is the coordinator of the ACAP study. And then of course, I want to thank the other ACAP sites and collaborators and trainees who've contributed so much to this large project, the funders of the project and of my lab more generally. And finally, I want to thank you all for your attention today.
Thank you so much, Dr. Lebel, for this great talk. And we now move to the Q&A phase. Just like to remind the audience to please address your question either using the Q&A, the chat, or raise your hand. And while people are typing, I'll use the benefit of asking a question or two. I think you basically demonstrated how you can use the diffusion parameters to predict the persistence or the lack of it of the symptoms. And I was wondering, just to entertain or to understand better, are there any immediate clinical implications on how to treat better or not according to this early knowledge?
Yeah, I think that that's a really good question, and it's been something we've been thinking about a lot. Of course, it's the hope, right, that we find some sort of biomarker that we could actually use or recommend clinically. At this point, there's so much heterogeneity in our data and so many kind of other factors that are coming in that we're certainly not there yet. But obviously it's a good goal. Yeah.
Okay, thank you. And next question from Doug. Can you please unmute?
Hi, thanks for a great talk. My question is slightly not entirely dedicated to the main thrust of your talk, but I was curious about your using combat and whether you actually, like if you ran your results all the way through all these analyses that you did, would you come up with different conclusions? Would things be stronger or weaker if you didn't use combat?
So that's a really good question. So far, we've only tested that explicitly in the CONNECTOME data that I showed. And so in the CONNECTOME data, we didn't find any differences between the orthopedic injury and the brain injury groups at any of the harmonization, like with or without harmonization. So that stays the same, but it's absent. It's hard to draw conclusions from that, which is why we looked at the age-related changes. And so that's where we compared the pre-harmonization and the post-harmonization, and we see much stronger relationships with age post-harmonization. So I think it's making the changes we want to look at more apparent, the harmonization is.
Thank you.
Thanks. And another question regarding on the context of motion. I was wondering what are the guidelines regarding sedations, or is it even possible to consider in these kind of ages?
So we don't sedate for any of our pediatric imaging, and most places don't because of the potential side effects, right? Those side effects outweigh any of the benefits for research. Obviously, if you're scanning clinically, you might consider sedation. Although as an aside, we've been doing a lot of work at our site to facilitate scanning of younger kids and kids with developmental disorders for research. And the clinical side of the scanning here is taking note, basically, and sort of realizing that we maybe don't even need to sedate or anesthetize a lot of the kids for clinical scanning. So we've been doing another project I can talk about some other time, looking at whether there's a need for sedation in clinical scanning of young kids. And overall, we're finding that most kids do fine without it. So, you know, subtle motion is a problem, and the subtle motion is probably not going away no matter how much we train or pack those kids in there. I think we need to find good data ways of dealing with it.
Thank you. Another question I see is, I think in the first few slides, you've shown that you've also tried a different metric. So was it inhomogeneous MT? I think you applied to this. If you could elaborate, or if you saw anything interesting.
Sure.
Using this.
So we only have that in our typically developing controls, which is probably the slide that was on. So in my study there, we've been looking at white matter development across adolescents, and we've been using a lot of different white matter metrics. So unfortunately, we don't have the IHMT in the brain injury kids. There just wasn't quite enough time, and as much as I advocated for white matter, I'm, there's other people who want other sequences in there. So we see, we do see age-related changes in IHMT in our typical kids, more so at the younger ages than at the older ages, of course. But we weren't able to look at that based on brain injury.
Great. So I think we can now conclude, and thank you once more for this great talk. And we hope to see you next time in person.
Great. Thanks for having me.